So for this last bit of the workshop, I thought, why don't we download the network from our FunCoup web server, and then assuming that we have a set of genes again that we want to analyze, that you might have gotten from your experiments, we can run another tool that has been mentioned but not shown so far because it's not integrated in the FunCoup resource online yet, which is TOPAS. It's a tool that we can use for detecting network models. So we can you can download the network, the FunCoup network, and input your genes together with the network into the TOPAS method, and you can retrieve the model, the network model for the set of genes. And then once we have the model, which is a subnetwork of the network, highly interconnected subnetwork of the FunCoup network, we will perform enrichment analysis here running ANUBIX, which is, again, as Dimitri already mentioned, the algorithm running behind PathBix. And we will compare its result to what I call his score here, which is the GEA method mentioned in PathBix instead. And we will do this by using the geo functional database. And so one reason why you would want to run our tool, network-based tools, on Flyne is because, for example, we don't have geo available in PathBix currently. So the first part is a brief introduction in how to read a TSV file. So I might say that might need to say that this notebook is intended for those that have some programming background. And if you're interested in using our tools with your customized databases, then please contact us. We will be pleased to help you with that. So FunCoup can be the whole network of FunCoup can be downloaded from the downloads webpage. And you can either download the full network, meaning for each link, which will be a row of the Tableau file, you will have the logged likelihood ratio for each evidence for all the spaces in support for each link. Or you can download the network in a compact form, and that's what I've done here. For simplicity, I've downloaded this file from the downloads page for human, so Homo sapiens, and I'm importing this into a data frame in R. So as you can see here, this TIBO or data frame is a table with about 5 million rows. So the full human FunCoup network is made of 5 million links. And the compact format comes with the gene in the third and fourth column, and then we have the probability functional coupling score, which is a score going from 0 to 1, and the final Bayesian score, which comes from the summation of all the likelihoods, basically in support of the link. And but you can also install some graph handler for further network analysis and visualization, like iGraph. It's quite popular in R, but also exists in Python. And what you would do if you have a table in a data frame format, you would input that by using the graph from data frame function of the iGraph package. And what I'm doing here, I'm inputting the data frame, so the links plus also the confidence score, and I'm explicitly saying that this is not a directive graph. And what we usually also do, again, we filter out edges that have a probability functional coupling score of less than 0.8. So we keep all the high confidence links. And we can do that by deleting edges. And then you also might want to remove isolated vertices, though. There's going to be some genes that after filtering will have 0 links. You can do that by removing those with a degree, or the degree is a property of a node, and it's related to its number of links equal to 0. And now you can see that the iGraph object looks like this. It says it stores some information, like it's an undirected network. There's no arrow in this dashed link representation. It's made of about 13,000 genes, and with more than 500,000 links. And then it stores attributes for vertices, P, and edges. And you can see it has names for vertices and the probability function, Kaplan score for the edges. Once you have stored your network into a graph object, then you're free to use all sort of functions in the iGraph library to, for example, extract basic network properties and do some further, even more complex network analysis. And you also might want to compare your network with other popular networks. Like in this case, I'm importing a pre-computed table. But if you're interested into the code, I might provide you with I can provide you with R scripts that can extract all this information, for example. And one thing you might want to do is, for example, look at the degree distribution of your network to see if it has a scale-free property, which is a property characteristic of biological networks. So we now start talking about application of FunCoup, and we will start with the use of TOPAS, whose acronym stands for top-down attachment of seeds. So we refer to seeds as the starting input of genes. This can be disease genes, it can be drug targets, and you can input these genes together with the network to the method. And then once it finds the full seed network, it will take a user input parameter that we will suggest to be no larger than 3. And it will, accordingly with this input parameter 1, 2, 3, it will find new potential disease genes or drug targets related to the seed in input, and then it will allow a maximum number of connectors to be in a shortest path between any pair of seeds to finally find the final model, and this is what is going to give an output. To showcase this, of course, if you want to run TOPAS, well, you can have a look at the paper at this link, and you have to git clone the repository, which all our repositories are stored under the Sonam and Bitbucket directory. And here we're running TOPAS on a set of genes which is related to biliary liver cirrhosis, which is an inflammation condition of the liver. And I'm picking these genes from a table that that it's published and downloadable with the TOPAS repository. So once you have cloned your repository, you can find under the benchmark data folder the benchmark gene set table. And what I'm doing here, I'm extracting the gene set for, again, biliary liver cirrhosis, which is called liver cirrhosis biliary. And I'm extracting the genes that you can finally see here. So these are 22 genes that we will use as seeds to find the disease network model for the biliary liver cirrhosis. And in order to do this, we have to source or import the TOPAS function by running this line. And then we can simply run the function TOPAS, which calls, which takes in input the network. And here I'm again excluding, filtering out all those links which have low confidence. I'm inputting the seed genes, those 22 that I just showed you. I'm also using expansion steps equal to 2 as actually as a default parameter here. And I don't need to run this in multiple cores. You might want to do this if, for example, you're not filtering out no low confident links, or your seed gene set that's large. And in the paper, we refer to we suggest to use parallelization if your gene set is larger than 200 genes. And then I'm saving these results into the folder results of this notebook. So later on, I will read up from this file. This is looks exactly as the first FunCoup network file I show you. So it's a data frame where each row is a link, and I'm here extracting the genes in this model. And again, now what I'm doing, I'm trying to distinguish what was a seed gene and what is instead a connector, or like a new potential biliary liver cirrhosis related gene. And as you can see, TOPAS, which is developed to maximize the number of seeds in the output, retrieves all the 22 seed genes, but I also find 15 new connectors. And you might have noticed that the only when you download the network from FunCoup is the genes will show up in with ensemble identifier. Hello, Golnat, can I ask you to mute, please? So they will show up with ensemble identifier, and if you are interested in of course, use different gene vocabulary, then you can use, for example, the UniProt mapping file or whatever system you prefer to map this ensemble identifier into gene names or in trace identifier, whatever. And here and then you can proceed with visualizing your model as long as it's not huge. Otherwise, it would just look as a hairball. And here, for example, I'm using the graph plot functions to the iGraph plotting function to visualize the model and coloring seeds in red and connectors in green. And if you're more interested into the biliary liver cirrhosis model, then we use actually this example to showcase how TOPAS can also find biologically relevant models in the paper. So please have a look at the paper if you want to dig into it. And then finally, once we have our model, what we can do is we can perform enrichment analysis, which again is an approach for gaining insight into the significance of high throughput data. But yeah, I want to stress out that when you run enrichment analysis locally, well, you can select your favorite functional database, as for example, the Gene Ontology biological process gene set database that I downloaded from the MCDB website. And what I'm doing here is I'm importing this file that I just downloaded, and I'm converting the table into a data frame, filtering out also, for convenience now, a small or too big gene sets. And I can do the same thing for molecular function, of course, and cellular components section of gene ontology terms. And then I'm combining all into a unique data frame, keeping the information what's the ontology they come from. And as you can see, I'm using the UniProt mapping file here to map the gene ID or trace ID into ensemble identifiers. And then I'm simply here showing what is the remaining number of geoterms per ontology after also the filtering. And it's a big chunk of the gene ontology database. So we have about 4,000 biological processes, 500 cellular components, and 1,000 molecular functions. So I will hurry up. So again, no need of explaining what is ANUBIX, but if you want to run it, you need to clone it from the ANUBIX repository. And this time you can import ANUBIX library by running a library ANUBIX. You might want to pre-compute the number of network links to geoterms, meaning you want to run the ANUBIX links function to get the number of links that each gene has to each of the geoterm. And then you can use this multiple times as long as you're using the same setting, filter setting of the network and the same functional database, or for enriching your input set genes. And that's what we're doing later on. We are using we're computing ANUBIX for the biliary liver cirrhosis model that we found with TOPAS previously. And by running the ANUBIX constraint function, inputting, as I show you, the network pre-computed number of links to geoterms network matrix. Again, I'm inputting the network as a data frame. With in this case, I can also input the full network. Then ANUBIX would set a cutoff and would remove, filter out low confidence links. You can specify the course again, the number of sampling operation to build a new model for each pathway. And then, of course, the gene set and the pathways, which will be the model genes and the geoterms data frame. And I'm also, again, for the sake of comparison, ran the E-score, which is simply a modified Fisher test as it's run in David again. And there is a function under the CRC folder saved in the E's R script. And I'm running the function. And finally, just wanted to show you again is that we can compare the number of significant gene ontology enrichments between ANUBIX and E's with a bar plot. And here I'm filtering out, I'm actually keeping all the enrichments with FDR corrected p-value lower than 0.01. And you can see that ANUBIX was able to find about 160, 170 geoterms to be related to the biliary liver cirrhosis genes, whereas we couldn't find anything based on overlap. And this is the final result, like showing the top 10 significant enrichments. You can see that the overlap is quite low, yet the number of links can be pretty high. And what I just wanted to point out is that the majority of these enrichments are immunosystem related, which makes sense as biliary liver cirrhosis is an inflammation condition of the liver.